India with a first class honors degree in electrical engineering. Long back, computer science ಅಂತ ಇರಲಿಲ್ಲ, ಎಲ್ಲ ಇಂಜಿನಿಯರಿಂಗ್ ಡಿಪಾರ್ಟ್ಮೆಂಟ್ ಆಗಿದೆ. ಗೊತ್ತಾಯ್ತು, ತುಂಬಾ ಇದೆ ಈ ಡಿಪಾರ್ಟ್ಮೆಂಟ್. ಯಾಕೆ ಆರ್.ಎಲ್. ಕಶ್ಯಪ್ ಅಂತ ನಂಗೆ ಕೊಡ್ತಿದ್ದಾರೆ. ಹೌದೇನೋ, ಆರ್.ಎಲ್. ಕಶ್ಯಪ್ ಅವರು ನಮ್ಮ ಕಾಲೇಜ್ ಸ್ಟೂಡೆಂಟು. ನೀವು ಆಶ್ಚರ್ಯ ಆಗ್ತದೆ ಕಳೆದೆ, ನ್ಯಾಷನಲ್ ಕಾಲೇಜ್ ರ್ಯಾಂಕ್ ಸ್ಟೂಡೆಂಟ್ಸ್ ಅಂತ ಕರಿತೀವಲ್ಲ, for the National College Basuri, he was the first person to get the first rank. Kashyap, actually, such a brilliant person actually. Avarella electrical engineering department ಬಂದಿರತಕ್ಕಂಥದ್ದು. And he was awarded a British Chevening Scholarship by the Foreign and Commonwealth Office UK for a Masters in Electrical Power Engineering at the University of Strathclyde in Glasgow. He completed his Masters with commendable mention in 2004. He completed his Masters with commendable mention in 2004. ಎಲ್ಲ ಸುಮ್ಮನೆ ಕೂತ್ಕೊಂಡಿಲ್ಲ. ನಾನು ಫಸ್ಟ್ ರ್ಯಾಂಕ್ ಅಂತ ಅಲ್ಲಿ ಫಸ್ಟ್ ರ್ಯಾಂಕ್ ಅಂತ ಕೊಡೋದಿಲ್ಲ. ಎಂತ ಇಂಪಾರ್ಟೆಂಟ್ ಈ ಲೈನ್ ನೋಡಿದಾಗ. Completed his Masters with commendable mention in 2000. Following the completion of his Masters, he developed a keen interest in neuroscience and won a scholarship by the BBVA Foundation to work with Professor Gonzalo D. Colvazia and Pablo Verona at the Universidad Autónoma de Madrid. Having learned how to perform single unit recordings and calcium imaging in leech neurons, his internship became thoroughly enjoyable. In 2006, he joined the Blue Brain team in Lausanne, where he began work on his PhD thesis. The focus of the thesis is on modeling synaptic connections between neurons and on validating the model against experimental data. When he is not working in the lab, he enjoys reading autobiographies, traveling and blogging. And such a brilliant person is with us. On behalf of me and on behalf of you and on behalf of the institution, I extend a very cordial welcome to Dr. Srinath Ramaswamy. And now that there is open, in a few minutes we will start. Dr. Srinath Ramaswamy, I request you.
So, thanks for the very kind introduction. I don't know if I have to use the mic.
ಇದು ಕಾಲರ್ ಮೈಕ್.
You hear me if I turn on
ಕಾಲರ್
the
mic.
mic ಇದೆ ಕೇಳಿಸುತ್ತಾ ಇಲ್ಲ?
Don't make that.
Don't strain yourself.
No, I won't.
ಹೇಗಿದ್ದೀರಾ ಸರ್?
So, that was quite an introduction. So, I guess I have to talk more than you because I'm the speaker.
Correct.
That's a big challenge in itself. But another challenge is to ensure that I don't put people to sleep. So, and another funny thing is, it looks like I'm talking in my own backyard. So, but finally, maybe my parents will get to know what I really did and that I did something useful in Switzerland and that I wasn't wasting my time. Anyway, so I'll talk to you a bit about the Blue Brain project. So in Indian newspapers, I don't know. But nevertheless, so this Blue Brain project was started about 10 years ago in Switzerland by a very brilliant neuroscientist named Henry Markram. And the mission, the objective of this Blue Brain project is to develop a facility that will enable us to build models, digital reconstructions of brain regions and eventually the whole brain. So human brain project so the human brain project here has the grand goal of simulating the human brain in the next 10 years. So we are still a little far from there, but the Blue Brain project put together an infrastructure to carry out experiments from rat brains, use this experimental data to kind of create a bottom-up model of a part of the rat brain. So now we start by building a foundation, we erect the walls and then the roof. So something like that. That's called a bottom-up model, where you start from the very bottom level, like recording from nerve cells, and then go up further levels of complexity and then build a model of a so-called microcircuit of neurons. What it is, Santa, I will tell you later. So, a big success, a big breakthrough for the Blue Brain project came about just last month when we published a so-called mega paper in a journal called Cell, which is a prestigious journal in biology. So this is quite a world record for a paper in neuroscience, because this paper actually has 82 authors. So I'm one of the first authors. It has 4 first authors with my professor and 2 other brilliant colleagues, and 4 senior authors. 82 authors from about 15 labs across 12 countries. So this is really a mega international effort in neuroscience. So neuroscience, people don't collaborate a lot. The left hand doesn't talk to the right hand. Scientists like to do their own little things, publish their own little papers, become big professors, and then all their work is forgotten. So part of the work that we're doing in the Blue Brain project is to actually keep alive all this data, all the studies that have been performed in neuroscience over many, many years, and kind of consolidate it in a digital repository. So it's like a digital library of neuroscientific data pertaining to one brain region. So I'll try and then walk you through the various steps of what it means to reconstruct and simulate a part of the brain. So reconstruction anta nanu helidre, it's like, for example, we all know what this is. We all have an iPhone or some kind of a smartphone. So if you want to understand how an iPhone works, so you buy an iPhone, and hopefully a lot of us won't really break it apart to see what's inside. But then if you break it apart to see what's inside, you will see that this so-called iPhone or any smartphone, so it's composed of a circuitry with several thousands of transistors. All these transistors are somehow connected to each other, and they exchange signals between each other, act like switches, so on and so forth. Right? So then, to understand any system, you have to take it apart. You have to see what are the building blocks of the system, and then put it all back together. Right? So you deconstruct the system, see what the system contains, put it back together. So you deconstruct and then reconstruct, which is what we are trying to do, but not for the iPhone. We are trying to do something much cooler. We are trying to do it for the brain. Now start ಮಾಡಿದ್ದು ಅನ್ನೋದು ಸುಳ್ಳಾಗುತ್ತೆ, because this seminal work of studying the brain was started by this guy, Santiago Ramon y Cajal, almost 110 years ago, in an isolated lab in Spain. So Ramon y Cajal was the son of a poor farmer, but he was a very ambitious guy, a very brilliant person, who went on to do a degree in medicine at the University of Zaragoza in Spain. So what Cajal did, among other things, was he also ended up winning the Nobel Prize for medicine or physiology back in 1905. So Cajal was probably the first winner of a Nobel Prize in modern neuroscience. So Cajal won the Nobel Prize with another Italian called Camillo Golgi, who invented a technique to label nerve cells to trace their morphology. So morphology, if you see a tree outside, right? Let's say you see a mango tree. What makes a mango tree different from a coconut tree? Kannige henge kaaniಸುತ್ತೆ. So a mango tree is very different in its morphology, right? A mango tree looks different from a coconut tree because they have different shapes, right? So just because they have different shapes, when you look at a mango tree versus a coconut tree, you are immediately able to recognize the two, right? In a similar way, there are millions of such mango trees and coconut trees and orange trees and whatnot in the brain, right? And these different trees are the neurons in the brain, the nerve cells. Right? And so these nerve cells, the way they are connected, the way they look, the way they function, their activity, right, is very different in all of us here. So ನಾವೆಲ್ಲ ಕೂತಿದ್ದೀವಲ್ಲ, ಅಲ್ಲಿ ಅವ್ರ ಬಾಗಿಲಲ್ಲಿ ಯಾರೋ ಬಂದ್ರು ಅಂತ ಒಬ್ಬರು ನೋಡ್ತಾ ಇದ್ದಾರೆ, ಬೇರೆಯವ್ರು ಯಾರೋ ಮನೆ ಮೇಲೆ ಇವತ್ತು ಊಟ ಏನು ಅಂತ ಯೋಚನೆ ಮಾಡ್ತಾ ಇರ್ಬೋದು, ಇನ್ನೊಬ್ರು ನಾನು ಯಾಕೋ ಇಲ್ಲಿ ಬಂದು ಅಂತ ಯೋಚನೆ ಮಾಡ್ತಾ ಇರ್ಬೋದು. So then ಈ ಯೋಚನೆಗಳೆಲ್ಲ, these thoughts, right, these emotions that are all coming out are due to the activity of neurons in the brain, the way these neurons interact and exchange information. So what Cajal did was to actually make wafer-thin slices of brains. So, even though you can't do this for humans, I wish we could, but we can't. So what Cajal did was he made wafer thin slices of rodents, of rats, right? And then he used this technique that Camillo Golgi developed to label neurons, to look at their morphology, and drew these very beautiful pictures. So, what you see here is a circuit of neurons in the retina, basically, right? So this was probably the first ever analog representation of how neurons look in the retina. And then, I mean, let's just take a moment to appreciate the meticulousness and the painstaking effort that Cahal actually went through to draw these, right? I mean, these are such beautiful drawings. So all these drawings, they're all immortalized. You can find all these drawings in the Cahal Museum in Madrid. So very beautiful drawings. And then he also drew these arrows to show or to hypothesize, he didn't know then, that signals between neurons flow in some particular direction and that neurons exchange information, right? But, I mean, this was almost 100 years ago. Back then, Cahal did not have the tools to really ascertain if these neurons exchange information. So he just hypothesized that neurons exchange information. He didn't know for sure, right? So then he drew these very beautiful, intricate diagrams of neuronal microcircuitry for different brain regions. So what we see here is the so-called neocortical microcircuit that Cahal drew. So, now here on Modlo, you see all these different neurons. You see a B, you see a D, you see a E, I don't know where A is. You see a G, for example. So these are all the different trees in the microcircuit, like ಇವಾಗ ಬೆಂಗಳೂರಿಂದ ಮಂಗಳೂರು ಟ್ರೈನಲ್ಲಿ ಹೋದ್ರೆ ಒಂದು ಸಕಲೇಶ್ಪುರ ದಾಟಿದ ತಕ್ಷಣ ಎಲ್ಲ I mean in this forest region you see all kinds of trees, all kinds of morphologies, right? So in a way the brain is very similar to what you see outside your window from the train, right? So there are all these very fine structures, these beautiful cells, neurons that connect to each other and exchange information. So this picture is a drawing of the so-called neocortical microcircuit. So, what's the neocortex on there? If you look at the picture of the brain, right, you see all these folds on the surface of the brain, right? Bangalore road potholes ಹೆಂಗಿರುತ್ತೋ ಗುಡ್ಡ ದಿಣ್ಣೆ ಅದೇ ತರ the surface of the brain ಅದೇ ತರ ಗುಡ್ಡ ದಿಣ್ಣೆ ಇರುತ್ತೆ. So these are called sulci, and if you look at it more closely, white ಆಗಿರುತ್ತೆ. So it's called rather gray. So it's called the gray matter, right? So this part of the brain is what gives us intelligence, right? So it's called the neocortex because it's the most newly evolved part of the brain. Okay, neo is new in Latin, and cortex is the brain. So neocortex, right? And about 80% of the brain consists of this neocortex. And in neocortex, there are different regions that are responsible for processing visual signals, olfactory signals, or touch, or for example, other higher order cognitive processes like naven yochne marthivi, naven maatadtivi, naven nodbiಟ್ಟು ಅರ್ಥ ಮಾಡ್ಕೊಳ್ತೀವಿ, etc., etc. So all this happens in the neocortex. So the neocortex is really a very important part of the brain. So what Cahal did was he actually drew the picture of how neurons look like in this most sophisticated part of the brain, the most newly evolved part of the brain called the neocortex. Looks like I'm boring you. It was just a joke. I was talking about what is I mentioned neurons many times. So what actually is a neuron, right? So a neuron is a very specialized cell type. And why is it specialized? Because neurons process information, right? So then each neuron in the brain has a very specific morphology. So it's again like the tree analogy I gave you. So they consist of structures called dendrites, right? The ones in red here. So these dendrites are very fine structures that receive information from other neurons. So this is a zoom in of a part of a dendrite. So these neurons are called layer 5 pyramidal cells. These are probably the most important cell type in the brain, right? They are very beautiful neurons. They have exquisite morphologies and they project to many different brain regions. And you will be surprised to hear that if someday I were able to pull out all layer 5 pyramidal cells from your brain, right? From Professor Ramchandra Rao's brain, right? The amount of fiber, these dendrites, right, that you see, if I were to pull out all the pyramidal cells in your brain, the amount of fiber would be so much that it would be you would be able to encircle the earth about 3 times. Just by one brain, right? Just one neuron type. So imagine the amount of fibers that's present in the human brain, right? So if I pulled out probably all neurons, then you might be able to make several trips to the moon and back just by using these fibers, right? So these dendrites collect information and these very fine structures called spines. And these neurons also have a cell body called the soma. So these dendrites receive information. The cell body somehow processes this information and relays this information through the axon, right? So in a way, this is like a post office, right? So letters cell birthday, and then you sort out all the letters, and then you make some sense out of all the letters that are received, and then it's sent to the different addresses, right? So that's basically what a neuron does. It receives information from all other neurons from different parts of the brain, which are all processed here, and then there's some sense that's made out of all this information, and then this information is relayed to other neurons through another structure called the axon, right? And so this is to give you an idea for the kind of diversity of neuronal morphologies that are there in the brain. So all these are neuronal morphologies from the rat. So you see there are so many different kinds of neurons in the brain, in the neocortex. So this is just a very small picture. So the actual numbers are even more mind-boggling because there are so many different brain regions. This is just a very, very minute snapshot of the kind of neurons that are actually there in the brain. So, and these neurons they're not solitary beings, right? So they like to interact, they like to communicate, they like to connect with other neurons. They are constantly making connections with other neurons, constantly exchanging information and communicating, right? So then this is an illustration of like a jungle of neurons that are constantly exchanging information. So how do these neurons exchange information? So these neurons exchange information in these structures called synapses, right? So the synapse, the word comes from Greek. So synapses means to touch in Greek. So to touch and form a contact or a connection, right? And so that's what you see here. So at these synapses, there are electrochemical signals that are exchanged from one neuron to the other. And that's how the signal that's sent by one neuron is received at another neuron through these synapses, right? And if you look at two neurons in the brain, like two neurons in the same brain region, you'll see that they are connected by many different points of contact, many synapses. On average, there are about 5 to 10 synapses that neurons use to connect with others. So basically, so you know what's a neuron, and you know what neurons do now. So then what Cahal did was he reconstructed the microcircuitry of neurons in the neocortex, and this is just to make you understand what's the neocortex. And as I told you before, so this is the illustration of a rat brain, and this is the entire rat brain, and this structure here is the neocortex. So this is the visual cortex, this is the auditory cortex, and this is the somatosensory cortex. So somatosensory cortex, if you see, if you look at the face of a rat, it has these whiskers that are constantly moving, right? That we use our hands to sense the environment, to touch, to feel, to know what we're touching, etc. Rats use these whiskers to make sense of their environment, right? So they constantly move their whiskers to try and sense if they're touching a block of wood or if they're touching a piece of cheese that they like to eat, so on and so forth. So when these whiskers move, all the signals, so these whiskers are like mechanical transducers, right? So what these whiskers do is they convert mechanical energy into electrical energy. So you touch something, that's converted into electrical energy, and these signals are transformed by these neurons to make sense of what they are touching, and that's how a rat knows if it's touching a piece of wood or it's cutting a piece of cheese, right? So then, what we did in the Blue Brain project was to actually study in detail this part of the rat brain called the somatosensory cortex, where neurons are arranged in this circular columnar arrangement, right? So when you look at the neocortex of a rat under the microscope, you will find that neurons are arranged in these so-called columns, right? So there's a microcircuit of neurons that are arranged in these so-called columns. And if you look at one of these columns more closely, you will see that it's like a forest, like an ecosystem of different kinds of neurons, different trees, mango trees, orange trees, coconut trees, whatever, that are constantly interacting with each other. So, in order to study this neocortex, what we did was we put together a process to reverse engineer the rat brain. So, what you see here is actually a cartoon of a rat with its whiskers, right? And then this is the gruesome part, but unfortunately science can be gruesome and scary. So we had to decapitate these rats. So we had to cut off their head under a guillotine, like they used to do back in France during the days of the French Revolution. To punish people, they would cut off their heads through a guillotine and they would decapitate them, right? So they would be severed, the head would be different from the body. So that's what we did in our lab, and we had to decapitate like thousands of rats, pull out their brains, and make wafer-thin slices of these brains to actually study the somatosensory cortex. And these wafer-thin slices, so of course, I mean, the brain is a biological system, right? So, the brain is located within the skull, which is floating in the so-called cerebrospinal fluid. The cerebrospinal fluid provides nourishment to the brain, it keeps it alive, right? So, naturally, if you remove the brain from its natural habitat and make slices of it, you have to keep it alive somehow. So you have to keep it alive in the cerebrospinal fluid, but you cannot extract the cerebrospinal fluid from the brain. So what you do is, you again study the composition, the chemical composition of the cerebrospinal fluid, and then make an artificial solution that's very similar in composition to the cerebrospinal fluid, and then keep these slices alive in this solution in a Petri dish, right? So this technique is called in vitro in Latin, because in vitro means in a dish. That's to keep something alive artificially. So then you keep these slices alive in vitro in a Petri dish. And this is more of a zoom in of the area that we are interested in, that we've been studying for many different years now. So this is a microcircuit of neurons in the somatosensory cortex. And these neurons have a very interesting arrangement in that there are about 6 different layers where you find different numbers of neurons in this microcircuit, right? So they are of course numbered from 1 to 6. So layer 1 is the most superficial layer, right? So if you look at like the brain from outside, what you actually see, all these folds, these potholes like on Bangalore roads, so that's layer 1, right? And then you go deeper and deeper and deeper, so you have a layer 2, 3, a layer 4, a layer 5, and a layer 6. And then the next is called the white matter. So roughly from mouse to man, if you look at microcircuits of neurons in the neocortex, they all have such a layered arrangement. So you pretty much find about 6 layers, neurons occurring in 6 layers, regardless of whether you're studying the mouse or the monkey or the man. So then, we kept alive these microcircuits of neurons in the artificial cerebrospinal fluid that I just told you, and this is a snapshot of how this slice looks under the microscope, because these neurons are very, very tiny, right? So you have to use a microscope to visualize these neurons. And then this is more of a little movie. So this is a sliver of the rat brain, like a slice that we procured, right? These slices are very thin. They're about 300 microns thick. So, I mean, one strand of the hair is about, I don't know, like maybe a micron. So, these are probably even much smaller than even a strand of hair, right? So then here, I'll play this movie again. So what you're seeing here is a slice that's being kept alive in a dish, and the experimentalist, the person who's going to record from these neurons, is using a microscope to actually look at these neurons, and this is the surface of the brain, this is layer 1, and then you go deeper, layer 2, 3, layer 4, layer 5, layer 6, you go deeper and deeper, and you see many, many neurons. So all these bulges that you see, these are actually the bodies of these neurons, the cell bodies of these neurons. And so I mean, we use these powerful electron microscopes to actually look at these slices. And what we did was something like Kahar did. So we injected a kind of an ink into these different neurons, right? And what this ink does is, of course, it flows through the whole morphology of a neuron and stains it, right? It's like painting a neuron. So what you see here is an ink that was injected into a neuron that has diffused through its whole morphology, and it has stained the morphology of this neuron that appears yellow under a microscope. Okay, and then these slices are further processed to reconstruct, to look at these morphologies in more detail. Right, so it's like, so you have this whole labeling of a neuron, it's like staining a neuron, and then you look at one of these neurons more closely under these powerful microscopes, and then you trace the morphology of this neuron. You look at how this neuron appears. I mean, is it like, what tree is it in the brain, right? So that defines the morphology of a neuron. So what Kahar did was he drew these neurons by hand, by looking at them under the microscope. But so we are about 100 years beyond Kahar. So given our technological capabilities today, we can do the same thing that Kahar did. So we took over from where Kahar stopped and created digital representations of these neurons. So Kahar drew these under a microscope with pen and paper. So we drew these neurons with powerful computers to trace their morphologies. And of course, since Kahar had to do this under a microscope with pen and paper, he had very limited resources, right? So he couldn't draw much. He was just one person, a one-man army. He couldn't draw much. So thanks to supercomputers that are available today, so we were able to start off from where Kahar stopped, and we figured out that the neocortex of the rat contains about 55 different neuron types. So it contains about 55 different trees, right? So this piece of tissue that's kept under the microscope, it contains about 55 different trees that come in different shapes and sizes that interact with each other in very intricate and different manners. Okay, so the neurons are interesting for two reasons, because they have a structure in that they have some kind of an interesting appearance, and they're also interesting because they have very specific electrical functions, very specific physiological functions, right? Several years ago, this technique called electrophysiology was developed to study the electrical activity of these different neurons. So this is one such electrophysiological setup that we have in our lab back in Switzerland, where we use glass electrodes to impale these neurons. So it's called the whole cell patch clamp technique, where you have a glass electrode that's filled with a conducting solution, and you inject a metal electrode into this glass pipette with a conducting solution, and then you go close to a neuron, as is shown here, and then you suck the membrane of the neuron, you rupture it, you apply pressure, you rupture it, and that way you gain access to the inside of a neuron. And that allows one to characterize the electrical activity of a neuron by rupturing it and by gaining access to the insides of the neuron. So this technique, electrophysiology, helps us to eavesdrop on the language of neurons. So it's like, I mean, if you have heard of the Tower of Babel, right? I mean, legend has it that in the Tower of Babel there were many different languages that were spoken. It was a small lighthouse kind of a tower where not many people could fit, and yet there were many different languages that were spoken. So in the brain it's very similar to the Tower of Babel in that there are these neurons that come in all shapes and sizes that also have a language of their own. In a way it's like India. We have people coming from all states and regions who look different and who also have their own language of communication. Right? So using this technique of electrophysiology, we were able to understand how these neurons talk, how do they communicate with each other, what kind of signals they generate, so on and so forth. Right? And using this technique of electrophysiology, we were able to determine that there are about 11 different electrical types. So there are 55 different morphological types, and there are about 11 different electrical types in the rat brain. So to further add to the complexity, a neuron of a given type can actually express many different electrical types. So that makes matters even worse, right? So as it is, to look at the morphologies of neurons, it's quite a tremendously daunting task. And then to add to it, there's another dimension of complexity of electrical types. And to add to a further level of complexity, you have the same kind of neuron, but which under different circumstances can use very different languages to communicate. Right? So we really have to map out all these dimensions of complexity experimentally and collect all this experimental data from the rat brain. So, further to add to this complexity, neurons also use these very different languages to communicate with each other. So, this is an illustration of an electrical recording between two neurons that are connected to each other. So, what's typically done here is you inject a stimulus into one neuron here, let's say neuron number 2, and then you record the activity that's evoked upon injecting this stimulus in another neuron, that's neuron 3. So they have very different languages of communication to add to the complexity. So they look different, they use different languages to talk to themselves, and they also use different languages to talk between neurons from other brain regions. Right, so I mean, this is what I meant by saying that neurons use different languages to talk to other neurons. So, I mean, this is a bit detailed, so I won't go here. So, yeah, it's sufficient to say that neurons use different, it's like a Morse code, right? So, they use a different Morse code to communicate with neurons from other types. So, this movie is a bit interesting. So,
creating realistic simulated neurons involves a series of steps from the lab to computer modeling. The first of which is to acquire a brain, a rodent brain in this case, slice it up and then navigate under a microscope to a relevant brain area. Once a relevant brain area is selected, using other techniques it is possible to target specific cells to patch.
So as you see here, these are the different neuron types that are being recorded.
The targeted cell is patched using a glass pipette. Once the cell is patched, there are two outcomes.
So here you're looking at the languages that are being used by these different neurons.
And the second is to fill the cell with dye. Electrical behavior of more than one cell can be recorded at a time depending on how many cells are patched with the pipette. In the LMMC, there is a patch rate that can patch up to 12 neurons simultaneously in slice. Dyed cells are visible under the microscope for 3D reconstruction. Every visible aspect of the cell is traced along the Z, Y, and X axis. Lab data is funneled into modeling workflows that will recreate and simulate both the
So then what we did there was, so we gathered experimental data. I'll just play this again. So we went from the lab, from gathering experimental data, to creating models of these neurons. Right? So again, in case people didn't understand what the lady was talking about, so what you see here is a slice, a rat brain slice. And here you're looking at the different neurons. All this is under the microscope. And here you're using a glass pipette to gain access to one of these different neurons. So you gained access to this neuron here, and then you apply these different stimuli to that one neuron to record the responses in all the other neurons that are surrounding this one neuron, or the neighboring neurons, right? So this is quite an intricate process, so we need to use these amplifiers that actually amplify signals that are gained at the level of microvolts to make sense out of them, right? And so this is the experimental setup that we actually use to gather all the data. And this is the staining, as I mentioned before. So you're injecting ink into this one neuron, a dye, that allows you to draw the morphology of this neuron, right? So once you gather the data on how this neuron looks and what language it uses to communicate, you can use this data to then create a model, right? A digital representation of what you see in the biological system. You identify certain parameters, certain features from the experimental data, and you use this data to build a model, a digital representation of what you see in the biological system. So this is what we mean by reconstructing the brain, in that you look at the biological system, you understand it, you understand the principles, the fundamental rules underlying the system, and then you extract numbers from this data and use these numbers to build computer models. So we call this this is something like an imitation game. Who's real and who's a replica? Does anyone know who's real and who's a replica? Can anyone guess? And? Well, that's what I said. Actually, both are replicas. So this is a wax statue of Amitabh Bachchan at the Madame Tussauds in Sydney, and this is his wax statue in New York, at the Madame Tussauds in New York. So both are actually replica here, but that's not what we are trying to do. What we are trying to do is a biological imitation game, right? Where we are looking at a biological system, doing experiments to characterize, in this case, the electrical behavior of neurons, right? And using all this data to recreate, right, the similar diversity, but in a computer model, right? So this is the biological imitation game that we are actually doing. So then, of course, another important step in this biological imitation game, which is very painstaking, is that you have to, well, as I said before, this neocortical microcircuit has 6 different layers, right? And these 6 different layers have their own thicknesses. So these are the thicknesses of these different layers that have been painstakingly measured under the microscope. And these are the number of neurons that are there in these different layers, right? So then we actually had to map out all this in order to build a model, and then we also had to map out what's the composition of these different neurons. Like, for example, you had to go to each of these layers and we had to say, oh, what's the number of mango trees in this layer as against the number of mango trees in this layer? Does this layer have more coconut trees compared to this layer, right? And so we had to do all this experimentally to map out the diversity of neurons across the different layers. And then we use these numbers to recreate the exact microcircuit that we saw in the biological system, but in a digital model, in a computer. Right? So this is no trivial task. I mean, I cannot do this through my laptop here, but we need very powerful computing resources to do this. And what you see here is a supercomputer that we use to do this. This is a supercomputer, an IBM supercomputer called the Blue Gene. So some of you might be wondering why the Blue Brain project is called the Blue Brain project. That's because we use a Blue Gene supercomputer provided by IBM to build digital models of brains that we procure data from rats, right? And the reason it's blue is because if you Google IBM and look at its logo carefully, you see that the B in IBM is blue, right? So that's why it's blue. It's called the blue gene. And why the blue gene? Because the first generation of the supercomputers was used to understand the human genome. So there was another big project about 1.5 decades ago in the US called the Human Genome Project, where they were successful in mapping out the genome of the human species, right? So they were successful in identifying the RNA and the DNA composition in the human genome. So I don't know what I did here.
Plus one a bit.
Lose contact.
So
Yeah, plus one. Off.
okay, look. Okay.
Predicting.
Right. So you see that the supercomputer is like several refrigerators that you find at your home stacked together. But I mean, each of these refrigerators has about 20,000 processors. So each of these refrigerators is equivalent to about 20,000 such laptops, right? So overall, the supercomputer has about 65,000 processors. So you're actually recreating this very tiny part of the brain. I mean, it's so tiny, it's probably the size of a sand grain. And to recreate this, you're using such a massive supercomputer with 65,000 processors. So that's quite mind-boggling, right? So another reason why we utilize the supercomputer is also to come up with an algorithm, right? Like a computer program that's used to derive or to tell us how these neurons are connected to each other, right? So previously I spoke of, maybe I should go back to that slide. So this model, this microcircuit of neurons that we're creating has about 31,000 different neurons, right? So they are in all shapes and sizes. So these structures called dendrites are very different for each of these different neuron types. These structures in blue, that's hardly visible here, called axons are also very different for each of these different neuron types. So what these neurons do is, I mean, they send their axon very close to the dendrites of other neurons and form these synapses, right? And so what we calculated from this supercomputer was that putting together 31,000 neurons, like 31,000 digital neurons in the supercomputer, we estimated that there are about 40 million such synapses, right? So it's quite a mind-boggling number. And there's no way we could have calculated the fact that there are about 40 million synapses. I mean, this is not possible to measure experimentally, right? So just imagine, as it is, it's extremely difficult to record from a few neurons. But imagine if you had like 40 million synapses in a piece of the brain that's the size of a sand grain. It's virtually impossible to measure that experimentally, right? So then we had to use the supercomputer in the model to come up with an estimate that there are a prediction that there are actually 40 million synapses in this part of the brain, right? And so here we are actually walking through this reconstructed part of the brain, right, to look at these different balls, these red and blue balls, which are the so-called synapses. So each of these red balls are, so there are two kinds of synapses in the brain. So there are excitatory synapses and there are inhibitory synapses. Each of these blue balls are excitatory synapses. So what do these excitatory synapses do? As is self-explanatory, these excitatory synapses excite other neurons, right? So then they somehow have these electrical signals that excite these other neurons and make these neurons generate electrical signals. Whereas these blue balls that you see, these are called inhibitory synapses. So their function is to inhibit electrical signaling in these other neurons. So if these neurons, if these excitatory synapses make the neurons go crazy by generating electrical signals, these inhibitory neurons calm the neurons, right, by inhibiting the generation of electrical signals. So you would be surprised to hear that about 85% of the synapses in our brain are actually excitatory. Just about 25% are inhibitory. And yet, well, I don't think most of us are calm and composed, but some of us are. So it's the inhibitory synapses that are more interesting to study in the brain rather than these excitatory synapses. Right? So then, I mean, this is a very simple cartoon of what we do with all the experimental data that I just spoke to you about to reconstruct digitally a part of the brain. So we map out all these different trees like in the Amazon forest, and then we characterize all these trees into a coconut tree, a mango tree, or an orange tree, and then we map out where these different trees sit in the jungle of the brain, right? And then we map out how these different trees interact with each other, what are the languages that they use to communicate within themselves across different trees, and then we have this virtual, this in silico. Well, so I mentioned in vitro. In vitro means keeping something alive artificially in a dish. In silico means keeping something alive in a computer, right? Because a computer has silicon chips, transistors, and in silico basically means keeping something alive in a computer, a model. So then, I mean, this is quite a jump that we go from a biological system, the biological imitation game, something that's very real, to something that's synthetic, which is like a digital replica of a living system. So, I mean, these are just some numbers. So, the piece of the brain that we are trying to model it's very small, as you see, it's just about 2 millimeters thick. And it's about 0.3 millimeter cube in volume. It's 1/3 of 1 millimeter cube. So it's really a very, very tiny piece. It's so small. It's like a grain of sand. Right? But this structure that's as small as a grain of sand has about 31,000 neurons. And I mean, these neurons are all very closely packed, right, in this sand grain-like structure. So there are about 110,000 neurons that are actually packed in this sand-like structure, and these are different numbers. The ratio of excitatory to inhibitory synapses that I just spoke about. And as I mentioned before, this tiny piece of this tiny sand grain has about almost half a kilometer of axons, of fibers, right? And about quarter kilometer of dendrites. And now, I mean, just imagine if I were to somehow model the whole brain, then the amount of fiber, as I said, would be so high that you would be able to encircle the equator probably twice or thrice, or even make several trips to the moon. So this is the process that we actually developed in a nutshell, in that we use experimental data, we identify fine principles and rules from this experimental data, right, like numbers basically, and then we extract certain parameters from this bunch of experimental data and use these parameters to build algorithms that then helps us build this part of the brain on the supercomputer. So this is what the reconstructed piece of the brain in a computer looks like. So I call this the in silico brainbow. So, you know, it's like a rainbow in the brain because you see all these different colors. So it's like a portmanteau of brain and rainbow. So it's a rainbow of neurons, right? And you see this spectrum, this colorful big gaillard of neurons in the brain. And then again, so we built this, but what fun is it just to build something, you know, and not see what it does, right? So then what we did was, again, we went back to our favorite supercomputer, and then we turned on the switch, right? So we have a model, and we ran this model on the supercomputer. We simulated this model, and then we saw that the way this model behaves is very similar to the kind of electrical activity that's generated in a piece of biological tissue, right? It was very, very similar. I mean, this was such a great emotional moment for us because to get to this stage, I mean, okay, this movie lasts 10 seconds, but to generate this movie, it took 15 years of work, right? So, and yeah, so, I mean, this is a small step for neuroscience, but a giant leap for mankind, right? So, I mean, we were really blown away because it performed exactly the same way as a biological piece of tissue would. I mean, that was such a great moment for us. And so, I mean, we would also like to share all this fun with you. So, what we did was, we made all the data, the experimental data, and this model, this nice simulation that I just showed. And all this is actually okay. So, all this is actually available online through a portal that's called the Neocortical Microcircuit Collaboration Portal. So, anyone can go to this website here, and then you can actually look at this microcircuit of neurons. We call this the digital reconstruction of neocortical microcircuitry. So you can go to this website. You can actually interact with this microcircuit. You can look at the different neurons, their shapes, their electrical activity. You can go to the different layers, the 6 different layers that I spoke about and all that. You can actually interact with this, right? And anyone can do this, anyone who's interested. You just go here. If you're interested, just copy this internet URL and then go to this portal and have fun. So, on this while I bored you about speaking of how we reconstructed a piece of the brain that was about the size of a sand grain, right? This is a very small part. But then the next steps that we are working on is actually to reconstruct the entire mouse brain, right? So this is a billion times a grain of sand, right? So we actually then reconstructing a billion times more neurons than what we actually have. So this is about 50 billion neurons. So we're not there yet. So I mean, though we are very ambitious and fast, technology is not as fast as us. So the technology to do this is still being developed. And hopefully in the next couple of years, if I visit here again and give you a similar talk, then I'll be able to talk to you about what we did in reconstructing the whole mouse brain, right? So this is just about the mouse brain, but I mean, these are just noise, very illegal. They're not very interesting, right? So none of us like it. So then the next step is to go closer to an animal that's not very far from us, or a distant cousin, probably a primate, a chimpanzee or a macaque, and to do the same, to reconstruct the brain of a chimpanzee. The even grander step is to someday reconstruct the brain of a human, which is the so-called human brain project, which is just started. So, but again, this is such a huge challenge, because as you see here, the order of complexity in terms of the number of neurons, how they are connected, how they are arranged, it increases exponentially as you go from amphibians or reptiles like crocodiles to insectivores, to rodents like rats and mice, to carnivores like cats, primates like monkeys, and finally humans. So you see, it's like an exponential leap, right? So it's very complex. But we're getting there. We have to start somewhere, so this is the starting step. So finally, so dhanyavadagalu and thanks to everyone. So it was about 1 hour.